Okay, so this is the last video of chapter 16. When last we talked, we were talking about G protein couple receptors. The general principles of G protein couple receptors is that the G protein couple receptor gets activated by binding to its ligand, causes a conformational change, which is going to cause the activation of the G alpha subunit. The activation of G alpha subunit is going to cause the GDP to be exchanged for GTP. Once it's GTP bound, it's going to become active. It's going to no longer be bound to the beta gamma, and the beta gamma will now be active. The alpha and the beta gamma can now do downstream effects. The ones we specifically looked at in the last video where the beta gamma was able to bind to a potassium channel in the heart muscle in response to adrenaline, acetylcholine, bind to it, the potassium channel, and the potassium channel then opens, and in response to acetylcholine, the heart slows down. We also saw the G alpha subunit in response to its ligand. It got activated by exchanging its GDP for GTP, and activated in one case the PKA pathway. The PKA pathway was the G alpha subunit activated an adenylene cyclase, and the adenylene cyclase changed ATP to cyclic AMP, and the cyclic AMP removed the inhibition from PKA, and PKA then had downstream effects. One of the downstream effects it had was to, for instance, activate an enzyme that activated another enzyme that gave you more glucose in response to adrenaline. Another thing that it did was PKA could enter into the nucleus and phosphorylate Krebs, which binds to the promoter of a gene and upregulated gene transcription. So understand the general principles of the PKA pathway and the specifics of the PKA pathway, and understand the general principles of G protein couple receptors and the specifics of G protein couple receptors. Another pathway we looked at was the PKC pathway. Just to remind you, that was that the activated G alpha subunit caused the inositol phospholipid to be cut. The diacylglycerol and IP3 were separated. The IP3 went and gated open a calcium channel on the ER, releasing calcium, which could do two things. It could signal downstream calcium signaling for many biological processes, or it plus the diacylglycerol could activate PKC, and PKC is a kinase and can have downstream effects, either activating enzymes or changing cytoskeletal elements, or upregulating gene transcription. And so understand the general principles of G protein couple receptors. Understand the specifics of the PKA pathway, the PKC pathway, and calcium signaling. G protein couple receptors just have, because of the many different receptors and many different G alpha subunits, beta gamma subunits, and then all of the downstream effector proteins of these pathways, have a lot of diversity in cells. They act quickly, they act for a long time, and they can really do a lot of the physiology. So now you know some of the molecular mechanisms for some of the physiological things that you've seen. Okay, so now let's move on to a different kind of pathway entirely, and that's the receptor tyrosine kinases. Receptor tyrosine kinases, in general, you've got two proteins. They're transmembrane proteins, and they are separate from each other. But in response to a signal molecule binding to them, very often a growth factor, this growth factor binds to them and brings them in close proximity to each other. Once they are near each other, they can cross-phosphorylate each other on a tyrosine. That's why they're called receptor tyrosine kinases. And the phosphorylated receptor tyrosine kinase very often acts as a scaffolding molecule for some enzymes, to bring that enzyme in close proximity to its target. And that's one of the things it could do. It could also act as a kinase of downstream targets, like in the case of Janus kinase. So the one we're specifically looking at is the MAP kinase pathway. So the growth factor binds to the receptor tyrosine kinase. The phosphorylated receptor tyrosine kinase, after cross-phosphorylating each other, the phosphorylated one acts as a scaffolding molecule through an adapter protein to bring the RAS activating protein in close proximity to the inactive RAS, which is a membrane-bound protein. Inactive RAS has a GDP bound to it. The GDP is exchanged for GTP once the activated RAS activating protein causes the exchange of GDP for GTP. The RAS is now active. And what does active RAS do? Well, it is the very upstream portion of a pathway we call the MAP kinase pathway. So MAP kinase pathway is a series of proteins. So I didn't name these, so don't blame me for this, but activated RAS protein activates MAP kinase. And as its name implies, it is a kinase that phosphorylates MAP kinase. And as its name implies, it is a kinase that phosphorylates MAP kinase. And as its name implies, MAP kinase phosphorylates MAP. And so many of the downstream things that are going to go on is we're going to activate enzymes. We are going to phosphorylate cytoskeletal elements. We are going to phosphorylate and therefore activate transcription factors, and we'll have cellular responses downstream of the effector proteins. So understand the general principles of receptor tyrosine kinases and how they work, and in specific understand the RAS pathway, the MAP kinase pathway, the inactive RAS exchanging GDP for GTP, becoming active, and phosphorylating the MAP kinase, which through a series of cascading phosphorylations causes changes in cellular behavior. So between G protein coupled receptors and receptor tyrosine kinases, this is a lot of really good stuff that you need to know for especially Chapter 18 and Chapter 20. Chapter 18 being cell cycle and Chapter 20 being cancer, because these pathways are involved, and understanding this later will just say activated, and you'll need to kind of have a feel for what could have been the upstream thing that activated means, because when these pathways break down, either constitutively active or an inactivator being knocked out or something like that can lead to cancer. So these are good things to learn and remember. I know I showed this slide before, but it bears repeating that there's a lot of crosstalk between different pathways. For instance, both receptor tyrosine kinases and G protein coupled receptors can activate phospholipase C, or maybe calcium activates protein kinase C as well as CAM kinase, or maybe the downstream target of MAP kinase from the RAS pathway has the same target gene as PKA, or there's a lot of crosstalk, and this is by no means an exhaustive list, this slide. There are many different pathways. I mentioned Janus kinase a second ago. Janus kinase, we're not going to go into the specifics of, but there are many different pathways besides PKA and PKC. So I'd really like for you to try to understand the general principles as well as some of the specifics of pathways that we've mentioned, and really commit these to memory. One that I like to end on is nitrous oxide signaling. So in response to acetylcholine in endothelial cells, arginine is broken down, and a byproduct of it is nitric oxide. And nitric oxide, because it's a gas, remember back to the membranes chapter, this gas can freely diffuse across the membrane out of the endothelial cells and into muscle cells and bind to its target protein, which is a guanylate cyclase. Guanylate cyclases turn GTP into cyclic GMP, and cyclic GMP can gate open a sodium channel. And so in response to nitric oxide, we have vasal dilation. The smooth muscle goes from contracted to flaccid, and so in response to nitric oxide, the microvasculature microvasculature becomes flaccid, or the lumen becomes larger, or something we call vasodilation, and so more blood flow through there. And it turns out that the little blue pill that is famous for older people is a inhibitor of something called phosphodiesterase, and phosphodiesterase cuts cyclic GMP. So if we're making cyclic GMP at the same rate, but we're not taking it away anymore, we get more cyclic GMP. So an inhibitor of phosphodiesterase causes a change in the microvasculature. Nitric oxide should have caused vasodilation, but here we get this the inhibitor of it, and so vasoconstriction, and so what we end up with is the side effect of the little blue pill that caused it to be a blockbuster drug. Okay, so that's it for cell cycle. Cell signaling, and I'm glad that we've gotten this far in the semester. Like I said, this chapter 16 was one of my favorite chapters, and it is going to lay the groundwork for our final push into cell division and cancer. Okay, thanks a lot.